Hi, I'm Olivier Siberio, CEO at Sensin US, and welcome to the Nutriscope calibration program. So today the purpose of this set of video is to present you with Nutriscope, the technology, what is the calibration, how do we calibrate Nutriscope, and the step-by-step process of calibration. I designed this video in a set of short video, so I would encourage you to watch all of them once, and then during the calibration project, to watch the ones that are the most relevant to you. So just for this introduction, we'll talk about two things. What is Nutriscope very quickly? We will have a demo right after. And what is a calibration program? So Nutriscope is a handheld scanner developed by Sensin that measure plant health and nutrients directly in the field. It helps farmers and crop advisors make better decisions by measuring in real time parameters such as pH, redox, breaks, nutrients, nitrogen, potassium, etc., etc. Every parameter needs its own specific calibration, and that's what we're going to talk about. The way it works is actually quite simple. Inside Nutriscope, you have a small spectrometer that emits light in the near infrared, visible, and UV. The light is reflected back by the leaf, and Nutriscope uh, analyzes this light using artificial intelligence to measure any of these parameters. Um, the calibration has to be done for Nutriscope because Nutriscope does not measure by itself, it needs to learn. And for that, we need your contribution. We will ask you to collect data points that are used for the calibration. Each data point is one set of scan and a corresponding lab analysis report. Once we collect all this information, we at Sensin will train models based on this scan plus lab report. We start with a data sample of 500 data points to build our first models, but every year we contribute again and we add new data points so that we improve the model. We'll have a section about this process of collecting 500 data points and reiteration process in this presentation. But that's all for now. The next step in our presentation will be to do a short demonstration of Nutriscope capability so that you can understand what is the purpose of Nutriscope and why we do all that. See you later. So how does Nutriscope work? It's actually pretty simple. So when you acquire Nutriscope for the first time, we'll need to create an account that will give you access to a mobile application and access to our Sensin portal where you can look at all your data. I will jump directly now after the account creation to the scanning process. So when you open your iPhone application, you have access to the screen, and the first thing you need to do is to connect your scanner to the phone. So you go to the screen scan, as I'm here now. You turn on the scanner on the back. You have a button that you hold it for a few seconds. When the scanner is on, you have a blue light that is blinking. You search for scanner, and the scanner number will actually appear on the bottom. You click on the number, and after a few seconds, the scanner is connected and the blue light is steady. Now you are ready to scan at this point. So I will do a single scan because we are doing a demo, but you have to know that in the field, not during the calibration, but when you use Nutriscope to monitor your crops, you have to do 15 scans to get to the nutrients. So just a demo, we'll do it. So the way it works is that you select the plants that you would like to analyze. So in this case, it's a lemon tree. You select the leaf you want to study. You open the scanner. You clip the scanner onto the leaf. It's non-destructive. Oh, I'm trying to get the focus a little bit better. And then you launch a scanner. So as I say, it takes like 6-7 seconds to do one scan. If you have to do, oh, we'll have a bad scan. So that's okay. Bad scan. That's actually one of the sectors. So one of the security of the system is that if there is a light infiltration, you can get a bad scan. So let's start again. So you say yes, you select the leaf, you clip it, and you take a measurement. So this time I will not unclip the scanner, but it's good like that. You can see it in action. So it takes 6-7 seconds to do one scan in the field. When you monitor your crops and you do 15 scan, you have to count 2-3 minutes of scanning time. But voila, you get your scribe. Once the scan is done, you can unclip the scanner. It's non-destructive again. You put the plant aside and you have access to the data. So the first screen that you have is plant nutrients. So we have like 16 nutrients already available for most of the crops. You have to check on a website which measurements are available for each crop. And the purpose of our calibration today is actually to get as many nutrients in this list as possible. So the reference data for the training is SAP analysis using a laboratory located in the Netherlands called Nova Crop Control. So when you look at this result, look at it as if it were a SAP analysis report. Then the second screen that you get is redox, so or plant health. So these constitute 4 parameters: pH, Brix, redox, and conductivity. This is a different set of calibration. So it's not included in today's scope. It could be a project for, a second step once we complete this calibration, but this is not yet included. But it includes, this framework pH redox that enable us to calculate the stress on the plant. But I won't expand. We have documents on our website that can give you more information about this framework. And finally, we have a decision support system, DSS, that use the data you just scan, ask additional questions such as the stage of growth of the plant you are considering and the condition of your root system. And from that, we'll actually make some recommendation. Today, the decision support system is only available on wheat and corn, and we don't how can I put it? It's not that we recommend you using it. It's more that it's a way for us to showcase the capability of Nutri-Scope. Because now that you have access to all this information all at once, and you can have as many scans as possible, well, you can develop your own decision support system. You can understand better your crop, and that's really what we would like to reflect with this kind of validation, demonstration of decision support. So this is for the demo. The next section will start to dive into the calibration. So hold on, because that will become technical. So let's dive into calibration. The calibration process consists in 4 steps, but the first step, data collection, is by far the most critical and the one that relies essentially on your contribution. So when we'll start the project, we will send you 10, 20, 30, 40 bags coming from the lab that we will use for the calibration. Each of these bags will have a unique identifier called the lab ID. When you will start the process of data collection, when you will start to work on one bag, we will ask you first to enter the lab ID into your mobile application. Once you enter it, you will be allowed to do the scan that corresponds to this bag. Each bag, depending on the crop, will require a certain number of leaves. So for example, in the case of blueberry, 30 leaves. That means that when you collect the data, you will need to collect 30 leaves of blueberry, and then you will scan each one of them individually. So 30 leaf, 30 scan. You place the leaves into the bag, you seal the bag, and you are done for now for one bag. Then the next step will be you collect another bag, you enter the new lab ID, collect 30 leaves, scan each of them, place in the bag, and seal. That's the first step. We will, in the end, collect the scan ID, and we will collect the lab ID, and that will enable us to make the correspondence between lab ID and scan ID, which means correspondence between the scan and the lab result. This is the most critical link that we need to preserve and protect during this calibration program. The second step is that sending all these samples to the Netherlands is quite expensive. So what we want you to do is to not do one lab analysis and send it to the Netherlands. That would make the whole project prohibitively expensive. So what we would like you to do is to collect somewhere around 8 bags. And once the 8 bags are ready, you box them and you place them and you ship them to the Netherlands in express mail. The box that you will use will be provided and the shipment fee will be covered by the project. Then the third step is the matching and the data cleaning. This is a step that we at Sensine will conduct. We'll look at each of the scan IDs as scan result. The lab result will first see if there is a good match between scan and lab result. Then we'll dive deeper into the scans themselves and we'll evaluate the quality of each of these scans. So if the scan is not properly executed for any reason, it may be a default of the system or a problem of light contamination, that's something you cannot see, we will eliminate these samples. Once we have a set of data, clean data samples, we will do the last step, which is the training of the model, which means that we'll look at these 500 data and train using machine learning a model to predict each of the measurements analyzed by the lab. So if we are successful at the end of step 3, our goal is to have 500 data points. But you have to understand that these 500 data points will never be able to reflect all the possible conditions that your crop can be exposed to, whether in your field or outside or in other location or with different cultivar. So we have like a larger domain of uncharted data of conditions that we will not be able to see, at least in this first iteration. But at least we'll have a shorter domain of 500 data points where we can do some work. So what we will do here is that out of these 500 data points, we'll blindly select 100 data points that we will use later for evaluation. We will use the 400 remaining to train the machine learning, which is a process where we'll calculate a model that based on future scan results will be able to predict some measurements. Once we completed this model, we will use the 100 data points that we just selected to calculate, to evaluate the validity of these models by comparing the model's result using the scan from the 100 to the lab result of this same 100. So these 100 serve as a blind test of the model. When the results are okay and with correct accuracy, we publish the data. Sometimes we estimate that the results are good but not great. In that case, we'll write this data as a draft or better. It's just a way to be cautious that it's good enough to be published, but you will have to be more cautious and we will improve that in the future. So that's the second step. That's why. But this validation only works internally to this 500 domain. When you have a growers and you have a specific field, you may be in a case where part of your of the conditions seen by your crops is within this domain, the 500 that we already evaluated, and maybe part of it will be outside. So if your crop are slightly outside of the initial 500 data points, we will lose accuracy. And the issue, and this is a limitation of machine learning that is very well known, but if you are outside, what will happen is that we will lose accuracy and we will not know why, and we will not even be able to tell you that we are losing accuracy. So the first model, even if we are using your field to do the calibration, may not be perfect. So what we do is that every year we gather new calibration data and we retrain our models. So that by expanding, by gathering new calibration data, we little by little expand the domain covered by Nutriscope so that at some point your field will be completely included and will be able to predict all the case, and all other fields also will be included. We believe that we'll still have some edge cases, so rare case of stress or whatever condition that may be unique to your system or to the season or to the weather, but at least every year we will improve the model gradually. So why do I tell you all that? Because you have to understand that Nutriscope works best when the condition of your field corresponds to the condition it has been calibrated to. So if you're outside, you will lose accuracy. So when you do the calibration, it is critical for you to keep that in mind so that we can build the most diverse set of conditions, of data points, so that the range, the initial range of calibration data is the widest and the broadest possible. So this will define the strategies that we'll use during the calibration. So what is this strategy? Its strategy is in two forms. The first one, at the bag level, we want you to tell one clean story. By that I mean that when you will collect the 30 leaves, we want you to take the same type of leaves. We want either they are young or old or close to a path or a specific cultivar. We want each bag to tell only one story. If you collect different type of leaf, different cultivar, the result of the scan will be mixed. The lab result will be also very mixed. We'll not be able to understand really what's going on. So one bag, one story. We want homogeneity in the bag. Now, the second part is multiple bag. When you look at multiple bag, we want each bag to tell a different stories. So maybe you will look in the first bag to young leaf, and old leaf at the second bag, or maybe a specific cultivar or a specific field. But the diversity will come from the story told by each of these bags individually. One bag, one story, multiple bag, that's when you get the whole picture, and that's where you expand the diversity. That's the key. You need to understand that when you do calibration. Don't mix different ideas in one bag, and don't build the same story across bags. So we are done for the calibration part of this presentation. But before we go to the next session, I just want to repeat the key takeaway because we said a lot today. So the first one is that Nutriscope learn by collecting data in the field. That's where your contribution is critical. Then, during the process of calibration, it is also critical for you to make sure that the lab ID is properly entered into the application. If we don't have the correspondence between the SCAD ID and the lab ID, we cannot link these two information, so data become useless. Our goal is to get 500 data calibration points after cleaning. If we get that, we will get a first model. The accuracy will be best within the condition provided in this 500 data point. We will lose accuracy if we go outside. But every year, we will retrain our model by gathering more data. So the accuracy will slowly, step by step, increase. Now, what is also very important is that when you do the calibration, when you work on one specific bag, these bags tell only one story. So the same type of leaf goes to the bag. However, there is a different story across each of the bags. This is what will increase the diversity of our data collection. Very important. That's all I want you to remember for today. The next part of the presentation will be more about the practicality of how to do scanning, everything. So we'll go through the equipment that has been provided, and step by step we'll go through the process of collecting leaf, putting them in the bag, shipping, etc., etc. So much more practical. So I look forward.